talking about the humoral immune system, and we were talking about the adaptive immune system, and we said that that your humoral immune system is the part of, well, they're both adaptive, the humoral immune system and the cellular immune system. And we said that the humoral immune system is about making antibodies, it's about attacking or your body defending against invaders that are outside of your cells. We said that the cellular part of the adaptive immune system is about actually destroying cells that are infected or damaged. One thing that I want to mention really quickly is that not everything that gets in your body is going to cause you to make antibodies. So we refer to some things as good immunogens and poor immunogens. So really good immunogens would be protein epitopes. So things like, if I take my red blood cells and put them in someone else and it's a different blood type, that's going to really stimulate a strong immune response. Because foreign animals or human cells that's really going to trigger your immune system. Those are good immunogens. Microbial cells and viruses, those typically tend to be very good immunogens, stimulating a strong immune response. Molecules from plants, if you have allergies, you probably realize that those tend to be fairly good immunogens. It's easy to have an allergic reaction to pollen. There are molecules that are poor immunogens, and usually they're logical. You don't want to be allergic to sugars, right? You need those for food. So molecules that your body needs, or that you might be normally found in food, proteins typically, just shorter polypeptides tend to be not so great as immunogens. So when we try to build antibodies, hopefully, if it's a pathogen, hopefully it's something that's a good immunogen and it's going to stimulate a significant immune response. All right, so reviewing just from last time, what an antigen is and what an epitope is. All right. Last chance to click in. So this is review from last time. All right, actually you guys did pretty well there. So an epitope is just a portion of an antigen, right? So if I have a protein that's from measles or polio or Bordetella pertussis, I might make multiple antibodies to the same protein that recognize different parts of that protein. And each of those would be considered an epitope. Oops, that was my yes or no. Okay. All right, antibody structure. Antibody structure. Okay, so here is the basic structure of an antibody. You may be familiar with this or maybe not, but I wanted to just point out a couple of features. So your body, over the course of your lifetime, makes millions and millions of antibodies towards different foreign items that it encounters over the course of your life. And all human antibodies have the same basic structure. So they have this stem region that, it says the C is for constant. So this is, all of your antibodies have the same sequence here. But what's different about them is this variable region. So the variable region changes depending on what it's binding to. And so we know that there were only 20,000 genes, and so you're wondering, how can we make millions of different antibodies with different variable regions, different shapes that are going to bind to different things that infect us? How are we going to do that if we only have 20,000 genes? And so there's a little video here that kind of goes through how this works, which is goes for the journal Nature. They're not slouches.
People are full of antibodies. A lot of antibodies. These proteins are a vital part of the immune system. But just how the body makes such a variety of them mystified scientists for decades. The human immune system has to fight off an endless variety of bacteria, viruses, and parasites. And for each invader, it needs a specific antibody. Luckily, it can generate billions of them, each slightly different, ready for any disease. But these antibodies, made from 2 light chains and 2 heavy chains, are proteins. And billions of different proteins should need billions of genes to make them. Humans have around 20,000 genes. The solution to this problem was a Nobel Prize-winning discovery. Antibodies are made by B cells. Like every other cell, B cells start off with the same 20,000 genes. But they also have an enzyme called RAG. RAG is a DNA shuffler. It targets the genes that make the antibody's binding site. There are 3 kinds of gene segments here, called V, D, and J. For the heavy chain of the antibody, humans usually have around 130 different V segments, 27 D segments, and 6 J segments. The RAG enzyme binds to one of the J segments at random, and then to one of the D segments, chopping off the spare DNA between them. Other enzymes then join the ends together. The same thing happens with a random V segment. So you end up with a gene composed of one V, one D, and one J joined together. This provides over 20,000 possible VDJ combinations for the heavy chain. The light chain can have another 400 possible gene combinations. Combining heavy and light chains gives approximately 8 million different possible antibodies, each with a different binding site. And that's not all. During the joining, extra DNA is added or removed at the junctions of the segments, which takes our antibody diversity from the millions into the billions. This is how our immune system can respond to diseases it's never even seen before. In fact, there's so much diversity that one of our antibodies could potentially bind to almost any target in the whole universe.
So pretty cool. You don't need to know the details of VDJ. I just want to get you to get the idea that in your existing 20,000 genes that the body makes these little rearrangements to create all these different possible antibodies. And the result is that any pathogen that your body gets exposed to, you have an antibody somewhere in there that can recognize it. And the key is for how your body finds that antibody and expands it in order to protect you during an infection. Question?
So if you have an antibody for something, is it just there and then it like gets more when you're exposed to it, or does it go away and then your genes memorize it so when it sees it again, it like makes them all over again?
So we're going to talk about how that works. So basically, once you build the cells that make that antibody, because your body is stimulated to expand it, those cells live in your body forever as memory cells.
Okay.
And they can at any time quickly start making antibodies.
Okay.
Questions?
Before that, are they just sitting around? And there's lots of them just sitting around?
And we're going to talk about that in a minute. So let me explain, because it's kind of an interesting process. Let me explain how that works, because you guys are asking great questions. You're getting ahead of where I am. So just very quickly, there's different classes of antibodies. The antibody that's most common that people are typically referring to are IgG, or immunoglobulin Gs. When your body first starts making antibodies at the very beginning, it always starts with an IgM and then it will class switch to IgG over time. So this is sort of the preliminary antibody that always changes to this one, or usually does. IgD is actually the receptor that's on the surface of a B cell. So we're going to talk about that. B cells, naive B cells have IgD on their surface, and that's what's going to tell that B cell that it should start secreting antibodies and become a plasma cell. Saliva, tears, breast milk, other body secretions produce a version of antibodies called IgA. And IgE is good to know about because this is the one that binds to mast cells and basophils and causes you to sneeze and have hay fever and allergic reactions. So antibodies, that general shape I showed you is how they all work. You can notice that general shape is present in all of these, but there's some different varieties of antibodies that have different roles in the body. So knowing just basically the names of these are IgD, M, G, A, and E and what they do. So this is associated with allergic reactions. This one is secreted. This is the one that's on the surface of the B cell. This is the very first one that's made after you're exposed, and this is the primary main antibody in your blood. So you should memorize that once. I know you won't remember it much past the test, but when you learn it again in professional school, which you will, you'll have this learned it once. Why do so many diseases such as measles occur only once in a person, yet others such as colds occur more than once? And that's actually a really important question. know the answer already?
Because, I don't know the word, but the flu, it changes every year, right?
That's right.
Yeah.
Yeah. So antibodies bind to proteins on the surface of pathogens. So if you're a pathogen like measles, and your surface is the same all the time, it never changes. It's like having one outfit on, and your immune system says, oh, I see, that's the measles in the red dress. And measles always wears a red dress, and so your immune system always remembers that it's measles. The flu virus, we talked about it being a master of disguise, and rhinoviruses that cause colds are the same way. They're constantly changing their outfit. And as soon as the immune system sees them again, they say, oh, you were in a purple dress last time, now you're in a blue dress. You must be someone else, I don't recognize you. Because they change their surface proteins. Okay, so bacteria and viruses that can change their surface all the time, that's a virulence factor to evade the immune system. That's actually a very effective one. Okay, so something like the flu is very hard to develop vaccines against because its surface proteins are always changing. Polio, measles, pertussis, a lot of other pathogens where we have really good vaccines, they look the same every time. And so one vaccine provides you with a much longer term protection. Yeah, the mutations are a big part. So how fast they can mutate and change is a big part of that. Okay. All right, so this is just, again, we're going to focus after this on how antibodies get selected, but just a little bit more about antibodies. You know, how do antibodies protect you? Well, they can do a few different things. So antibodies can bind to bacteria and cause them to clump. This is called agglutination. See how it's causing these bacteria to stick together in clumps? So if you're a phagocyte, it's like picking up a chip and it's got 3 or 4 other chips stuck to it, right? You can take a huge bite of a whole bunch of them at once. My food analogies are not working for you. It's almost lunchtime. This should work. All right, so this can make phagocytosis more infectious and also can neutralize these organisms. So down here, so it can cause them to clump. It can also prevent them. See, if they're coated with antibody, not only are they more delicious because they're opsonized, right? We've dipped them in the guacamole. Coating with complement or coating with antibodies, that's going to make them more susceptible to phagocytosis. So that's our dipping. But agglutination makes the process more efficient, and by coating them with antibodies, you can prevent them from interacting with the receptors on the cells that they might infect. So it also causes neutralization. So when bacteria or viruses get coated on the surface with antibody, you really can prevent a lot of problems from happening. You can prevent them from being able to attack a target and make them more susceptible to being destroyed. One of the things that can happen, we talked about last time complement just a little bit. We scratched the surface, and but I did mention the classical pathway, the alternate pathway, and the lectin pathway. Remember that briefly? I said those are just multiple ways complement can get activated. One of those ways is the classic pathway, and that results in antibodies binding to the bacteria. That actually stimulates the complement cascade in your blood. And then antibodies also, even if an organism is really large, antibodies can bind to organisms. So here's a worm, and you see the little tiny antibody on the surface, but it's actually recruiting a cell type called an eosinophil. And when the eosinophil binds to this antibody on the surface of the worm, it produces molecules that are going to help destroy this worm. So perforin and lytic enzymes to kind of try to basically douse that parasite in a nasty chemical bath to try to kill it. So here's a picture. This is kind of cool. So this is a little nematode larva, and these are white blood cells. These are eosinophils, and they're attacking it. Isn't that cool? Based on this, it's called antibody-dependent cell-mediated cytotoxicity, which is a mouthful, ADCC. Antibody-dependent cell-mediated cytotoxicity. So if you ever wondered how your body can kill a big worm with just cells, this is how. There's strength in numbers, right? Plus, remember, these cells are releasing these nasty chemicals. So that worm, it kind of looks like it's going to lose that battle to me. All right. So these are the mechanisms. You know, in a multiple choice exam, if I had, which of the following are mechanisms by which antibodies help your body combat an infection, these would all be different ways that would be the case. All right. Oh, this is just another slide on showing. This is another larva. This is actually a flatworm. And you can see the eosinophils binding to it. And so they're binding in response to the antibody, and then they're secreting all these nasty chemicals. Super cool. See the little epitopes, right? The antigens have epitopes. You can see the antibodies, a lot of the terms that we've been talking about, that's the constant region of the antibody, that's the variable region. I'm going to keep going. Are you ready for the next slide? Okay. So here's our white blood cell producing antibodies at the plasma cell, and he looks mean. The antibody hits the bacteria, and he's like, I'm hit. Come on without me. So he's going to get eaten by a phagocyte. Here's another one. This is when bacteria trash talk to antibodies. He says, oh yeah? These are the antibodies. You and what course of antibiotics? I think those are hilarious. Okay, so I like to start with a little discussion of antibodies. It's usually something you've heard of. And then the rest of this lecture kind of fits together. So remember that we said that there are, in the adaptive immune system, let's break this down. Let's start at the top. So we have the immune system. We said there was a first line of defense, which is your skin, secretions, stomach acid, right? Things that keep the bacteria from ever getting in. Then we said there's the innate, the second line of defense, which is when it gets in, you're born with some things that can immediately try to kill any pathogens that get in you, right? And we said overall that's the innate immune system, things that you're born with that were present at birth. And then we said there's a third line of defense, which is the adaptive immune system. So these are parts of your immune system, we said they're very slow. They can take a week to 2 weeks to really get going. But they are ways for your immune system to adapt specifically to a pathogen that gets in you. And antibodies are one of those ways. We said that you have all these different combinations of variable regions that can bind to any shape in the universe, right? So any pathogen that gets in you, your body can make antibodies against it. So we want to step back now and ask, well, how do antibodies get made? What is the process by which your body recognizes the pathogen and makes antibodies against it? And that process is very similar for your body actually making cells that will attack infected cells. And these are the two branches of the adaptive immune system. So you have lymphocytes, T lymphocytes, you have B cells and T cells. And so B cells make antibodies, that's what that is. This is an IgD, it's present on the surface of the B cell. And then you have T cells, and they have their own receptors that are also have the kind of variability that we were talking about in that little nature video. And it's the same, made the same way. So these have T cell receptors, and B cells have B cell receptors, otherwise known as IgD, which are these essentially antibodies that are on their surface. So this is going to attack infected cells and tumor cells. This is going to attack bacteria and viruses when they're outside your cells. Okay, so these are the two parts of the adaptive immune system. So B cells and T cells actually both start out in your bone marrow. They're actually both start as that hematopoietic stem cell that we talked about when we did the blood cell lab in class. And the B cells and the T cells go from the bone marrow and they mature. In the case of T cells, they mature in the thymus, and B cells remain in the bone marrow and mature in the bone marrow. And so you have this immature cells that mature and differentiate into T cells and B cells. At this stage, we refer to them as naive T cells and B cells. Virginal maybe is a word that would be easier to understand. What this means is they have their specific receptor on them, but they have yet to be stimulated into producing, cloning themselves and making more of them, or making antibodies in the case of a B cell. What they do after they're produced is they migrate to the lymph nodes. Remember we said the lymph nodes are like a checkpoint, and all your lymph fluid has to go through the lymph node and get screened. It's TSA, right? So these guys are like the TSA screening people. And what they're doing is they're screening everything in your lymph nodes, and they're looking to see if they can bind to it. All right, they're looking for things that they can bind to that might not be you, that might be hazardous to you. So as your lymph gets filtered through your lymph nodes before it gets dumped back into your bloodstream, it's got to pass through these guys. Okay? All right. So this is just the surface of a B cell. Remember, this is happening in the bone marrow. This is the surface of a T cell. And what I really just want you to see here is this is the IgD, or the B cell receptor, and it's attached to the membrane at this stage, and it's binding. It's like binding sites for pathogens or anything that's not self. And by the same token, a T cell receptor also has binding sites for anything that's non-self as well. Okay. There's actually one other thing that I want you to notice about B cells and T cells, and one of them is that, remember that we said that I think we said this before, that all nucleated human cells have an MHC class 1 receptor. We're talking about HLA receptors. So I was talking about self and non-self, and I said that all of your cells have proteins on their surface that just differentiate you from someone else. Right? And that we can't take my kidney and stick it in someone else because they will not recognize my proteins. So those proteins are, sometimes they're called human leukocyte antigens, they're also called MHC class 1 receptors. And they simply, that when your white blood cells are patrolling, they're looking to see if you have your white blood cell receptors. They're looking for, your nucleated cell receptors, these MHC class 1 receptors. So that's what this is. B cells, dendritic cells, and macrophages also have another special kind of MHC receptor, and that's called an MHC class 2 receptor. So I just want to point out that it's present on the surface of the B cells, but it's not present on the surface of the T cells. So MHC class 2. And I'll show you in a minute why MHC class 2 receptors are special, because they tell us something about, they have an important job and they tell us something about that cell type. All right. I'm just thinking if I want to tell the story with that. Let's do this one first, and then we'll go back and do this one. Okay. So all human nucleated cells have MHC class 1 molecules. So here's an MHC class 1. A non-nucleated cell would be something like a red blood cell. Okay, but all of your cells that have nuclei in them, which is most of them, have one of these MHC class 1 that's specific to you. And again, this is why we can't put my kidney in one of you, and vice versa, because your body would say, oh, that's I see MHC class 1, but it's not self, it's somebody else, so we're going to get rid of it, and your immune system will attack it. So this molecule is found in all nucleated human cells. Class 2 MHC is found on B cells, macrophages, and dendritic cells. And when a cell type, these three, has a class 2 MHC, it means that it's an antigen-presenting cell. Remember we talked about some cells having that special characteristic about phagocytizing bacteria and viruses and chopping them up and then putting them heads on stakes and feet on stakes and thighs on stakes, the whole thing, all the parts. So that's, the stake is an MHC class 2 receptor. Unless your job is antigen presentation, putting things on MHC class 2, then you don't have that receptor. So these three cell types, white cell types, are the ones that have the job. We say they're professional antigen presenters. That's their job. If you're a neuron, your job is a professional neuron. It's not a professional antigen presenter. If you're a kidney cell, you're a professional kidney cell. You're not a professional antigen presenter. So when we say professional antigen presenter, we're saying that this is presentation of antigen on the surface is actually their job. That's the reason they exist, is to present antigen to other cells in the immune system. Okay. So hold that thought because we're going to come back to it in a minute. Another term, because we haven't had enough yet, is something called clonal selection. And I want to talk about this now, and again, we're going to refer back to it. The video I just showed you basically explained how we can get billions of combinations of different variable regions on antibodies. Does that make sense? Billions of combinations? Okay. And they explained that it does this gene shuffling in order to do that. When you get a pathogen in you, how does your body know which of the millions of potential T cells and B cells, which are the right ones to expand up? And the way it works is like this. Okay, so here's our stem cell, and it's going to produce lots of different, let's say these are B cells that make antibodies. So we've got a B cell with this shape antibody and a B cell with this shape antibody. See how there's all these different ones? Okay. All right, so the first thing that happens is we know your body notices that some of these, because it's a random process, some of these actually will bind to your own tissues. And if you start to make antibodies or killer T cells that recognize you instead of an outsider, what are they going to do to you? It becomes an autoimmune problem, right? Your own immune system will attack you. So the first thing that happens is that we eliminate clones that are specific to your own proteins. And you're happy this happens. This is an important role that the immune system has, to eliminate things that would recognize self. Okay, so we want to clear out those cells that would recognize self. All right, once we've done that, then the rest of these are all good because they're all potentially cells that have antibody shapes that might recognize a pathogen, an invader in your body. All right, so here's our repertoire of possible shapes. And now, oh no, it's a bad guy, it's an antigen, it's just invaded us. Help, help! Our innate immune system isn't going to save us, it's not enough. So what happens now? Well, it turns out that as this gets brought to this particular T cell or B cell, do you see how this binds right into the little receptor there? And that triggers your body to say, oh, we have a cell that can protect us, and it divide, and it's going to divide into millions and millions of these little cells that travel through your body and get rid of all these guys. And that's called clonal selection, but maybe without as much drama. Okay, does this make sense? So it doesn't make sense to expand this one, right? Because it's not going to help you. So your body is basically figuring out which of the right, and of course we're only showing, what, 6 of these, but you have the millions of them, right? And it takes a while, I said it was slow, a week to 10 days to 14 days, to find the right cells that bind to the right shape and then expand them up so they can start helping you get better. So when you get sick, you feel sick for a while because your immune system is trying to find the right cells. It's like looking for your keys, right? The immune system is trying to find the right cell to help you get better, and it takes a while for it to do this because it's looking for a needle in a haystack, literally. It's looking for the 1 cell or 2 cells in your body that have the right shape to take out this pathogen. Okay. All right, let's do a couple quicker questions and then we'll keep going. Which cells have MHC1 on their surface? Last chance to click in. Okay, so most of you correctly saying all nucleated cells, and that's the correct answer. Okay, B cells, macrophages, and dendritic cells have both MHC1 and MHC2. They're nucleated, so they have the I'm self molecules, but they also have MHC2 to do antigen presentation. Okay. Which ones, I kind of just told you, see if this, asking it a different way. Which cells are professional antigen presenting cells? It's their job, it's what they live for. Last chance to click in. All right, and you guys correctly said B cells, macrophages, and dendritic cells. These are the professional antigen presenting cells. That's their job, is to present antigens. Okay, lots to cover. T helper cells. Remember that I said last time, I believe I said last last time, that people who end up with HIV, the infection HIV, they used to all die back in the 1980s when we first, pretty much the very few exceptions, and there are interesting exceptions, but with very few exceptions, they all were killed by HIV. And the reason is because that HIV infects the next cell that we're about to talk about, which is the T helper cell. And the most important cell in your immune system, they're all really important, but if you have to pick a most important, it would probably be the T helper cell, because it's really the linchpin of the immune system. It doesn't actually kill anything itself, but it's the one that stimulates the other cells to do their jobs. Okay? So let's skip the text and go right to a picture. All right. So when you first get an infection, the first thing that happens is the helper T cells are activated. Here's the process. And once you understand this process, you've got a lot of the immune system down. All right, so here's our bad guy microbe, right? We should have put an evil face on him, but he's kind of spiky, so you know he's bad because pointy things are bad, right? So this pointy thing gets eaten by this. The book said macrophage, but I added or dendritic cell because either one can do this job. So you see how it's getting phagocytosed and chopped into little pieces? Chop, chop, chop. And then we're going to put it on a steak. And the steak is an MHC class 2 protein, right? Antigen presenting cell, so it has MHC class 2. So this is going to fit on the steak, and then the steak is going to be put on the surface of this antigen presenting cell. Okay? And now this antigen presenting cell is going to find a helper T cell, and the helper T cell has to have the right shape. Remember we talked about clonal selection? So helper T cells are selected that way. You've got lots of different naive T cells, and you have to find the one with the right shape. So this is what's happening in your lymph nodes. And I always want to show my armpit because you have lots of lymph nodes in your armpit. Jim in your groin, all those great places. Okay, so the lymph nodes are here, there, or these cells are in the lymph node. Here's our antigen presenting cell, and it's presenting it to a T cell, and it's the right fit. It found the right one. It took 6 days, you've been blowing your nose and coughing, you've been miserable, but finally, the right antigen, the antigen has been presented to the right helper T cell. And now finally we're going to start making some progress getting better. Okay? So this causes this helper T cell to be activated. And what it does is that when this binding happens, it stimulates an interleukin to be made. So interleukin 2, that's going to cause the cell to begin dividing, which is what you want because it's the right molecule now. You want to make sure you have lots of it. And what it's also going to do is it's going to make other chemicals that are going to stimulate the antibody-producing cells and the killer T cells or the cytotoxic T cells. So when this cell gets activated, it makes copies of itself, and then it also makes molecules.